Hi class, welcome to lecture number 14. This lecture is on applications of immunity and it focuses on chapter 18 of the textbook. As I'm sure you're very happy to hear, this is also a very short lecture, and I like to include this lecture in our class because it informs two of the labs that we do. For our class, it's actually lab 15 and 16, I believe. You're going to do one lab, but they're both simulations, of course, because we're online. But lab 15, you're going to simulate doing a flow cytometry analysis of a patient's blood. And in lab 16, you're going to do an ELISA to determine whether or not your patients have, I think it's lupus that you test for. So only 4 learning objectives and 4 sections of the chapter to do for this particular lecture. And this lecture is all about immunity and how immunity works, like how we can define immunity, how we acquire immunity, and how we can manipulate our adaptive immune system, basically our antibodies, to basically into becoming little molecular tools. So let's talk about defining immunity first. Remember that your adaptive immune system has to be acquired. In other words, although you're born with all the parts, you don't have it yet, right? You need to educate your adaptive immune system, your B cells and your T cells, to get those memory cells and to differentiate between self and non-self and so on. So there's 2 main types of immunity, and that's active and passive. So I'm going to go ahead and skip this slide. You can obviously you have it, but I'm going to skip to the next slide because I like looking at the pictures while I talk about this. This is right out of your textbook, and there's 4 different types of immunity. We have active and passive. Again, so active and passive. Where's my where's my pen? We have active and passive here, and then we have natural and artificial. So natural active immunity, of course, is essentially the immune system that you get as a result of surviving and growing up, right? As you so much happens to your immune system when you're a baby and when you're a toddler, and that education is called natural active immunity. So as those pathogens and as those environmental antigens kind of go into and through your body, your immune system will educate itself based on what's dangerous and what's not dangerous. On the other hand, we can artificially introduce those antigens to your body to teach your body what could be dangerous. And of course, what we're really talking about here is artificial active immunity, better known as vaccination. So when we think of artificial active immunity, I want you to think of vaccinations. Remember, this is hopefully as a part of, if you're very lucky, of course, as a natural part of growing up, you're not going to encounter the tetanus toxin, for example, right? You'd be in big trouble if you did. But if you get the tetanus vaccine, you're artificially introducing a piece of that tetanus toxin to your immune system so that your immune system knows what to look for. And that, of course, will give you protection if and when you do encounter it in the future. And we're going to talk about vaccines and their types here in just a second. On the other hand, we have passive immunity. Natural passive immunity is the immunity that's given to you by your mother, either through the placenta in your via IgG antibodies, or during breastfeeding through IgA. So natural passive immunity is the immunity basically given to you by your mother, whereas artificial passive immunity, you probably haven't heard of this, but this is a very expensive type of immunity where you essentially are getting serum or plasma from other people, or sometimes even animals that have some type of protective component to them. So there's been a lot of talk actually about this with COVID-19, that the plasma of people that have survived a coronavirus infection, that their blood plasma or their blood serum contains is it plasma or serum? I forget. So they're usually used, those 2 terms are usually used interchangeably, and I want to say that one is in the absence of clotting factors or not. Oh, how does that work? So if you draw a patient's blood and you let it clot, the liquid portion is called plasma. And if you take a patient's blood and you treat it with anticoagulants and you spin it so that the cells go in a centrifuge so that the cells go to the bottom, the part that's on top is called the serum. So they're modestly different, distinct terms. You'll learn all about this when you're in nursing school. but you can basically get someone's serum or their plasma, and that can give you some protective, because that's where the antibodies are, right? So if they have really high antibody titer in their blood, by taking that into your system, you might be able to passively get that immunity from them. So it's an interesting avenue of investigation for COVID researchers right now. So let's talk about vaccination. We haven't talked about vaccination. There's a really great video that I linked to on the lecture page that you should watch about what immunization is and why it's so important. And there's also a assignment where you watch a movie, and it talks about the current controversies and kind of social issues surrounding vaccinations and so on. It's really hard for me as an immunologist, as a former immunologist, like I should say, and someone who's studied vaccines and studied the molecular mechanisms of vaccinations, to understand why people are afraid of vaccines. But I'm also not a parent, and I also understand that if someone is saying something could hurt your child, that sometimes science doesn't help very much, and that it's scary, right? And I have a lot of sympathy for people that have children, but and are scared about vaccines and are unsure. But there is no reputable source of information or research or science that backs up in any way, shape, or form a danger that vaccines pose to people. There are side effects of vaccines. They're extremely rare, and any kind of deleterious side effects, so any kind of dangerous side effects that could cause harm or even death, are extremely rare. And but they do happen. What we're talking about, an order of magnitude chance, just minuscule chances of side effects. So how vaccines, there's two main categories of vaccines. So there's attenuated vaccines and inactivated vaccines. So let's talk about attenuated vaccines first. So first, let's talk about characteristics of vaccines. Like, what should a vaccine do? Well, it should have few side effects. In other words, it should be safe. It should give long-lasting protection. Ideally, you should only have to get a vaccine once. That'd be, of course, be the ideal situation, and that provides lasting, lifelong immunity. And as a kind of bonus, like what we would ideally have in a vaccine, is that it would be cheap to make. It would be stable. In other words, it doesn't necessarily need refrigeration, or it can be shelf stable for a long time, or it could be stored in a powder type of form or something, and easy to administer. Nasal spray, an oral capsule, a shot that doesn't cause, any kind of adverse swelling or skin reaction, and so on. So those are the characteristics, right? If we wanted to describe our perfect virus, or perfect, not a virus, a perfect vaccine, these are the things that vaccine would have. Now, there's two types of vaccines that we can talk about, and they're attenuated vaccines and, like I said, inactivated or dead vaccines. So I don't think we call them dead vaccines. We call them killed vaccines. The antigen is in fact dead. And so a lot of people don't realize that there are quite a few vaccines. We're actually injecting ourselves, we're introducing to our immune system something that's actually alive. It's a microbe or a virus. So we're really talking about bacteria or viruses at this point. And in an attenuated virus, why do I keep saying that? In an attenuated vaccine, what happens here is we're going to introduce let's just say a microbe that is extremely modified, such that it's not going to do well in a human body. So the most common way that this happens is we take a microbe or a virus and we pass it, we propagate that virus or microbe in an animal, a non-human animal, a pig, a rabbit, a horse, something. And that virus, I don't know why I keep going back and forth between viruses and microbes. This virus, for example, will accumulate mutations that allow it to go to survive in a horse or a chicken or whatever it might be, but it won't do well in a person. So now when we introduce that virus to a person, the virus is really not functional. It's not going to work very well. It's not going to be able to get inside our cells. It's not going to be able to replicate very well. And our immune system will be able to handle it no problem. This is there's a lot of advantages to this kind of vaccine because it's going to do because it's alive and because it's trying to replicate and because it's trying to actually gain a toehold in our body. Our immune system's going to turn on, and it's going to take this thing very seriously, and it's going to mount a proper immune response, and therefore we're going to have long-lasting immunity to whatever antigens this invader, this vaccine, is going to have. The disadvantage to a live vaccine is that there is a very small risk, and I'm trying to think. I haven't looked at the CDC's website. There's actually there's a page on the CDC's website that you can go to that shows incidences of BAC mutations. So essentially, the bacteria, the virus, was able to actually mount an effective infection. So they track those, of course, across the country. And I can't remember the last time there was one of these. The chances of this happening are so small that they really just don't happen anymore. The technology with propagating these vaccines are getting better and better. So the chances, even though there's a small chance, there is a chance of this BAC mutation where the virus or the bacteria can change in such a way that it causes harm to the host. Now, let's contrast that with inactivated or killed vaccines. So in this case, the vaccination product, the thing that we're injecting into the person, is dead. It's no longer alive. It's killed. It's usually killed with a chemical, not by heat, because remember that heat distorts proteins and therefore will distort antigens. And we don't want that. So we usually, they are usually killed by something like formalin or some type of preservative that is going to chemically destroy the bacteria or the virus while not changing those antigens too much. So of course, the big advantage to this is that there's no risk of BAC mutation and that these are typically shelf stable for a really long time because they're dead. Attenuated vaccines, so living vaccines, they have to be kept alive, right? If you think about it, we can't just put a virus on a shelf and keep it there for years until we might need it. So we need to usually keep those refrigerated. These inactivated vaccines, they're usually in powdered form and we can rehydrate them as we need them. So the problem with these is that they don't mount a really good immune response. So because they're not trying to replicate inside, excuse me, because they're not trying to replicate inside our body, our immune system doesn't get turned on all the way, and therefore we need booster shots. These aren't as strong, so every so often we have to get a booster shot. A perfect example of this is diphtheria or tetanus. In fact, tetanus is a type of toxoid. If you recall, I mentioned this when we were talking about exotoxins. A toxoid is an exotoxin that has been modified so that the toxic part is gone, but the antigenic part is still there. So all that happens, for example, in the tetanus vaccine, the tetanus shot, they introduce the portion of that exotoxin that will tell our immune system what it is, but the part that's toxic isn't attached. And so it's it's a really great idea because it basically educates our immune system to be on the lookout for this tetanus toxin, but we can't get any type of toxic effect from it. There are variations on this so essentially what we can do is we can take antigenic portions from a virus or from a microbe and introduce that portion into our body, and that educates our immune system to that particular epitope or that particular antigen, but there's no way that these can be pathogenic because all of the harmful portions are removed. So a lot of times, at least in the past, inactivated vaccines or killed vaccines have been administered along with an adjuvant, but adjuvants have, I meant to look at this before I did this voiceover, but adjuvants have kind of gone by the wayside. They're kind of controversial because basically an adjuvant is a chemical or a substance that is injected with the killed vaccine that basically its job is to kind of piss off the immune system and create inflammation and turn on the innate portion of our immune system. So there's some really interesting crosstalk between, everyone always thinks that vaccines are about the adaptive immune system because it's all about antibodies and creating memory. But our innate immune system plays a pretty important role too, because it's going to inactivate those antigen presenting cells in order for our adaptive immune system to work. So the role of an adjuvant is basically to turn on our innate immune system so that we can get that inflammation and we basically get longer lasting immunity. They have a lot of problems with them though. They cause a lot of like injection specific problems. They're if you've ever seen it, I think it's smallpox. It creates that scar on people's shoulder. But there's a lot of problems associated with adjuvants. So they're kind of a two-edged sword. Double-edged sword? Double-edged sword. So here's a nice overview of the pros and cons of attenuated versus inactivated vaccines. And you can see a comparison between the two. We talked about because attenuated vaccines actually try to replicate, remember they're living but hamstrung viruses or bacteria that are injected into our system. Because of this, because they create that cell-mediated response, because they're trying to replicate in our system, it's going to fully turn on our immune system and we're going to get that cell-mediated response, and we're definitely going to get good IgG production. We undergo class switch because we're bringing in the full response of the immune system, so we're going to get not only IgG but also IgA. Long-term protection because we're creating those long-lived memory cells. We only need a single dose of these, and so on. Again, a couple of disadvantages is that we basically can't give attenuated vaccines to immunocompromised people. So if we don't have an immune system, or we have a very low functioning immune system, then the risk is simply too high for someone who's immunocompromised to get these attenuated vaccines. Remember that there is a small risk of mutation, so that the virus or the bacteria can become virulent again. And the other disadvantage, of course, to attenuated vaccines is that they have to be kept refrigerated, or they have to be kept cold in some way. And this is one of the big things with working with vaccines in developing countries, is how are we going to refrigerate these vaccines? It's a very, very big problem because of either no electricity or the electricity can go out. So if there's these kind of rolling brownouts or rolling blackouts in these developing countries and the hospital loses power for any period of time, all of the refrigeration goes and now all of their vaccines don't work anymore. So contrast that with inactivated vaccines, where their stability is very good because it's a product. It's not a living thing. It's a portion, right? It's that antigenic portion of a piece of something that used to be problematic, but we've cut out all that problematic portion. So there's no risk of back mutation. There's no risk to immunocompromised person, but the protection doesn't last very long. It's only short-term protection, and therefore we have to have booster shots every once in a while. Okay, so let's switch over to a completely different way of thinking. So we've talked about the types of immunity a person can have, active versus passive, natural versus artificial. And we've talked about the two types of vaccines and how they can help people by different mechanisms. First half of the talk. Second half of the talk is something completely different. We're now looking at antibodies as a scientific tool. So we can actually, because remember, the defining characteristic of an antibody is that it's highly, highly specific. So if we have an antibody and we know what that antibody is specific for, we can actually use that antibody to detect what it is that we're looking for. These are called immunoassays. Another word for this is serology or serological testing. So we can actually use these antibodies as diagnostic tools. And what we can do with these antibodies is we can label them. We can stick a fluorescent marker on the Fc portion of them, or we can stick a specific color of a molecule on the Fc portion, or some little marker, radioactivity, for example. And we can use these antibodies to target whatever that antibody is specific for. Another thing that we can do is we can use these antibodies to aggregate either cells or viruses or epitopes that they recognize. So here's an example. Here we have an organism, and we don't know necessarily what this organism is, But we know that we have some antibodies, like in a lab. This one is not necessarily in a person, in a lab that are specific for Treponema pallidum. This is the causative agent of syphilis. Now, so this is situation A. In another situation, let's say situation B, we have a patient's serum, and we don't know if this patient has syphilis or not, or have ever been exposed to syphilis. But we do have the syphilis, the bacteria that causes syphilis, this Treponema pallidum bacteria. So in either case, if we mix these together and we get a positive aggregation, in other words, these antibodies recognize this bacteria, we know, one, that this unknown bacteria, in case A, that this bacteria is Treponema pallidum. In this case, we know that this patient is either currently exposed or has been exposed in the past to this bacteria. So we can not only use antibodies to identify an unknown bacteria, as in the case in A, but we can also use the serology to tell whether or not a patient has been exposed to this bacteria, either in the recent past or in the distant past. Oh, you probably heard that. I'm so sorry. I probably just forget I just said that. It's very noisy in my alley. Hold on. Let me close my window. Okay. On the other hand, we can do an ELISA. So this is an enzyme-linked immunosorbent assay. This is actually what Lab 16 is for your activity. So imagine that we have like a tiny little plate, and we're going to attach to that plate an antigen X. And we want to know if, like for the lab that you're doing, I think you're looking for antigens that indicate the patient has SLE, systemic lupus erythematosus. So we're going to add some antigen. Let's say it could be an antigen that we know is indicative of the HIV virus, for example. So we want to see if a patient has antibodies specific for HIV. So we're going to put an HIV specific antigen, let's just say, for example, on a plate, and we're going to add the patient's serum to it. So in the top case, the patient's antibody is going to recognize that antigen. And so we can go through this process. We wash off any antibodies that aren't stuck to the antigen. And now we're going to add an antibody that recognizes patient antibody. And notice that that second antibody has a marker on it. And in this case, let's just say it fluoresces green. So if it's green at the end, we know that we have antibodies that are recognizing, in this case, HIV. This is a positive result. We know that this patient has IgG, in this case, against HIV. This patient is HIV positive. On the other hand, we could add patient serum to this antigen, and the patient's antibodies don't recognize antigen X, or the HIV antigen. So that when we wash this plate, the antibodies aren't bound, and therefore they're going to wash off. So when we add that second antibody, there's nothing for the antibody to connect it to, and we get a negative result. So this is a very, very common test, and it's very reliable, and many diagnostic labs rely on ELISAs to diagnose patients with either particular conditions or exposure to certain antigens. And so it's called an ELISA, ELISA, enzyme-linked immunosorbent assay. Here's another application. This is fluorescence microscopy. I included this one mostly because they're really pretty pictures. And what happens here is we can actually take cells, so we can grow cells in culture, in an artificial environment, and then we can treat these cells with antibodies that are either directly labeled themselves, or we can treat them with two antibodies, one that recognizes the cell or the cell component, and then the next antibody recognizes the first antibody. So either one works, so the direct or indirect. And what we can do is we can use these fluorescently labeled antibodies to identify particular parts of the cell. So here we can see that the green parts are, I'm pretty sure this is an antibody that's specific for actin. And so actin are these kind of cellular structures that move things around the cell. And the blue portion, what do you think the blue portion is specific for? Well, hopefully you recognize this as mitosis, and therefore the blue parts that are moving around are DNA, right? This is so this is the nucleus of the cell, and you can see this is what would this be? Early interphase, early prophase, late interphase, early prophase. Here you can see the chromatin is condensing, right? And here you can actually see the spindles are forming, the actin spindles are forming, and you can see metaphase, early metaphase plate forming here as the chromosomes are lining up. Here's a beautiful picture of anaphase, right? They're starting to pull apart. Here's telophase, and here's late telophase. So we have successful mitosis here. Pretty cool picture, all because of two fluorescently labeled antibodies, one that recognizes actin and one that recognizes DNA. Here we have another picture. These are, they look like HeLa cells. These are a type of epithelial cell. And you can see that we have, again, some type of nuclear stain, that blue stain that's staining the nucleus blue. I'm not sure what the green antibody is here, but in this case, the actin filaments are stained this red fluorescent color. And what I like about this picture is that it very, very clearly shows an apoptotic cell. So you can see that this cell here, you can see that the nucleus is being torn apart, right? There's lots of tiny little nuclear blebbing that's occurring, and it's shrinking, and we see this very characteristic round shape of an apoptotic cell. And we know all of this because we know how to utilize these antibodies and turn them into tiny little magnets that are very, very specific and attached to very specific parts of the cell, and then glow a specific color so that we can take pictures, and they turn out very pretty. Okay, so if you recall, let me just go back to this. So again, the two examples I just gave you, the fluorescence microscopy and the ELISA, are using this one here, the labeled antibodies to identify. But we can also just kind of very simply use antibodies to aggregate, to look for aggregation. That's what we did here, right? We can see that the antibodies are recognizing, in this case, Treponema pallidum. But we can use these aggregation assays to, for example, blood type. We already talked about blood type in the previous lecture, in lecture number 13. But we can type human blood groups, right? We have A, B, and O. We can type blood simply by the ability to aggregate with antibodies or not. So here, remember, we have our 4 different blood types. And then the blood type, of course, is determined by the type of antigen on its surface. And I think I just kind of included this because I think it's kind of interesting to show the distribution of blood types among different categories. And this is, of course, just in the United States here. But it's interesting to see just the distribution of the different blood types. I think it's kind of cool. I'm AB, actually, so I have a pretty rare, unfortunately, it's not a very good blood type to have because I have to be careful with blood transfusions. Okay, so how does this work, right? So imagine that we have, remember that IgM is a pentamer. You can see this little pentamer right here, and it's really good at agglutination. Remember that agglutination is the fancy term for clumping, right? Because it can link two things at the same time, right? Two different red blood cells at the same time. It forms these really nice aggregates that can become visible. So imagine that we have an IgM antibody that is specific for A, right? So this IgM can recognize the A antigen on the surface of red blood cells. If we apply IgM that is specific for anti-A to a patient's blood and it aggregates, and then we add anti-B and it doesn't, we know that this patient is type A. So we can actually use these antibodies to type the blood of patients if we don't know what their blood type is. And we can see, right, really, really nice, clear, very visible aggregation or clumping is occurring. This might be a new record, less than 30 minutes. Yay.